Okay. So this is going to be really quick. Okay. This is a short section of messenger RNA. Which DNA code probably produced it? So they're asking for the DNA, so just which DNA matches with it. GCC ATA. Did y'all get D?
Yes.
No. Oh, it's D.
Fine. Oh my God.
Okay, because look at GCC ATA. Okay? This one over here. Which of these represents the DNA segment from which this section of messenger RNA was transcribed? Which represents the DNA?
A.
A, C, T. Yep. DNA molecules separate into single strands, which then are used to construct two identical strands of DNA. This process ensures that daughter cells are genetically identical. Okay. The picture shows a person's karyotype. Which of the following can be best determined by this? The presence of an additional chromosome. Yep. Which of the following nucleotide base sequence complements this? So it's just which one binds complementary. TTA. Yep. If the template of the strand of DNA is this, what is the complementary? T, C, T. Yep, that's the only one. Okay, which of the following can be, we already did that one. So this is definitely DNA, and these are nucleotides. Okay. In order for DNA to replicate, the strand must separate at which of the following? It must separate between the bases. So it's the hydrogen bond between the bases. Between sugars and phosphate, no, that would be over here. Between the 8 sugar pairs, no. No. Between the 4 nitrogenous bases, that sounds right. Between only 2 chemical, yep, between the bases. That's the first step. Okay. When a strand slippage errors occurs, an insertion can result. How does this? Insert, added, right? Is that it? Right?
That's what happened.
Yeah. When insertion, and how does this? Yeah. Characteristics. Which components of DNA are referred to as the genetic code? So your code is the order of your bases. Nitrogenous bases.
So B is right.
Yes. Okay, this right here, I'm telling you right now, that is DNA because it's double-stranded. That is RNA. DNA to RNA is transcription. What are they doing? They're transcribing DNA to RNA. Preserving genetic information for future, and I don't know about that, deleting, nope. Transcribing information in the DNA, producing more nucleotides for DNA. They're transcribing, okay? A mutation that occurs in the gametes, the sex cells of the organism will be transferred to which of the following? If it's in the gametes, it's into your offspring. The offspring of the organism. Okay, this is shown below. So they're pointing to a bond between the bases. Bond between adjacent phosphate and deoxyribose, no. The hydrogen bond between 2 complementary nucleotides, yes, because technically that's a nucleotide and that's a nucleotide, and they're complementary, A and T. How does the DNA in cells determine organisms' traits? Why do you look like the way you do? Your DNA makes RNA, and the RNA makes proteins, and you look like the way because of your proteins. DNA contains codes for proteins, yes. Okay, how is information for a specific protein carried on a molecule? In the code, the order of the sequence. As a sequence of nucleotides, I think so. In the double helix shape, it's not because of the shape, it's not the ratio, and it's not the sugars and phosphates. It's the code of the sequence of nucleotides. If a genome is mapped of a bacteria species and is found that the DNA contains 30% of adenine, so if it's A, T, G, C, if 30 is for A, what must be for T?
30.
30. This is 60%, so between these has to be 40, so it's 20 and 20.
Oh, so I did get that right.
16, 20.
I did the math right for the first time.
Okay. The differences between two molecules include the type of sugar. What makes up such the molecules? These molecules are. The differences, the sugars, so the sugars are carbohydrates. Oh, wait, sorry. The difference between the two molecules include the type of sugar, and the molecules identify the 4 nitrogenous bases. Oh, okay. So it's nucleic acids. Okay, so they're talking about DNA. So they put a adult cell into a culture. Okay, let's see. Which conclusion is supported by the data in this investigation? The DNA information for development was silenced in differentiated cells. The transplanted DNA functioned as the original nuclear DNA of the egg. The DNA from skin cells and epithelial cells was not transcribed. The transplanted DNA mutated. All I can really tell is that when they did this, you know, this, whatever they got it from, acted as the DNA. I didn't really like that question. Okay. Okay, DNA damage can turn off genes involved in cell signaling pathways. Turning off these genes can cause less mature cells to divide too rapidly. Blah, blah, blah. What is it called when you divide too rapidly? Tumors, cancer. Good.
Well, I thought of cancer, but I was like, close enough.
Exactly, that is. Okay, the fact that a strand of yeast with a certain defective gene can use the human's version. Okay, this is on the STAR test often, guys. So guys, if the fact that a strand of yeast with the defective gene, so yeast, can use the human version. So if yeast can use the human version, it means that that gene for yeast and humans are similar, okay? So it's not because yeast and humans have the same food supply. They do share a genetic code. I wish it would have been for that particular gene because they don't have identical genomes. They don't have identical whole genomes, but they do have a small similar code. And they're not both eukaryotic. Okay, this right here is transcription, DNA to RNA. What is being produced? Messenger RNA. Look at, there's U's. Messenger RNA is being produced. Okay, C. The technique known as chromosome painting is the result of scientific research. Scientists use chromosome painting to mark the locations of genes on human chromosomes with fluorescent tags. It is also possible to apply this technique to chromosomes of many species. Chromosome painting allows for which of the following? Which I'll put? Or did you not get? Did you get that? Okay. So guys, if you had a chromosome, so here's. Oh my gosh, I don't know where the button went. I did something, y'all. Okay, so if here's a chromosome for the human, and here's a chromosome for the cat, and I said chromosome painting. Here's a fluorescent tag for the I protein on a human. Here's the fluorescent protein for the I protein on a cat. What can you do by knowing that? A comparison of the genomes of different species, the sequencing of proteins for many species, an increase in mutations, the extraction of amino acids. Guys, all you can do is compare. So the best answer is comparison. Shh. Okay, up here. Nucleotide sequence comparisons are possible because all organisms, guys, all organisms, look up here, have the same what? We don't have the same genes, but we all use the same for what? Good, so we have the same DNA bases. We don't have the same organelles, or it's not because of chromosomes or proteins, it's because we can share the bases. So guys, that's why if we eat strawberries, we break them down and we take that strawberry nucleotides and we rearrange them into ours. So my skin right here, originally, the cells of those, the cells of the nucleotides of the DNA in the nucleus of these cells came from what I ate. That's why it's important to eat healthy. Okay, come on, let's get to, oh, I like this one, guys. Okay, so up here, normal sickle cell. Okay, first off, if you ever get two codes and say, here's normal, here's sickle, okay, first thing you need to do is always count them. That is the easiest way to figure out what kind of mutation it is. So look up here. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. 12. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. Good. So if it's the same, was anything added? There was no insertion. Was anything deleted? It's not a deletion, okay? T, G, A, G, G, T, C. Ooh, here's the difference, okay? What type of mutation? There was not an insertion deletion. Did something shift completely over? It was a substitution, okay? Go behind the camera. Okay, go behind the camera. A mutation is least likely to affect a cell when the mutation, okay? This is what I talked about, where if you code for AAA is lysine and then have a mutation AAC, but it's still lysine, as long as it codes for the same amino acid, it's not going to do anything. So as long as it reverses the order, no, allows the total number of bases, no, replaces with a complementary base, no, produces a triplet that codes for the same amino acid. So that's when you have the same amino acid.
Where's your eye?
Yeah, the chair's over there.
Ooh.
Oh, my foot. Our video. Oh.
Ooh, my baby. Okay, guys, so practice this if you need more practice, or come in and see me. As you can see, all of these practices, I show you where the mutation is, and that mutation is usually in that codon.
Can I get changed?
Okay,